Hi guys, this is part 3 of the GI system diseases. We're going to start off with cholera here. So cholera is caused by a curved rod, a Vibrio cholerae or cholera, that is found sort of normally naturally in the waters. So we have Vibrio can be in fresh water and also marine waters. Because it's in the water, the shellfish can get colonized with it, as well as crustaceans. And then if we drink the water or if we eat the foods, the seafoods that have the cholera, we can get it. So infected humans can also be carriers of it. Not carriers of it, but infected humans can provide the passageway to infection. So transmission is either foodborne, so you eat the contaminated food, or it can be fecal oral because the bacteria will be found in the feces. And so the areas where there are cholera, sort of regular outbreaks, the Vibrio cholera is in the water. So those would be the more endemic areas for us. We are not an endemic area for Vibrio cholera. So when we experience outbreaks, it's mostly by our asymptomatic carriers. So cholera is a little wimpy, right? It's very sensitive to our stomach acids. So we need a really large dose in order to become sick with cholera. When the surviving cells enter the intestines, they attach to the walls and they multiply, but they're non-invasive. So cholera is not a bloody stool, but it is a massive loss of water. So the cholera toxin, which is the sort of nasty, virulent agent of this bacteria, really causes electrolyte loss, and then the water comes drawn out via osmosis. The next slide will look at that. Because the goblet cells are also losing their contents, the goblet cells will lose their mucus, and you'll have flecks of mucus in there, and that's why they call it a rice water stool, which is pictured down here. Sorry for that gross picture, you guys. And this really, this mechanism to lose water, where you have this osmotic draw out of your intestines, is very nasty. And it causes very rapid loss of water, which can lead to shock. We've talked about shock before. Shock is a very sudden drop of blood pressure from the loss of fluids. It can also be from the loss of blood, which is fluids, but you can either lose the water component of your blood or you can actually lose the blood itself. But both of those cases, you're going to have very low blood pressure. The blood's going to have a lot of trouble reaching your vital organs, and you can die from shock. And look how much, you have 3 to 5 gallons of fluid can be lost a day. That's a ton of water. And so this is what shock really does, right? You have the fluid loss, your blood gets thicker, and then your vital organs fail to function properly because your heart's not able to pump the blood there. Yeah, so it's very deadly. You can die in less than 2 hours from dehydration. So in terms of the treatment for cholera, it's rehydration first and then antibiotics, because the dehydration is what kills you. That massive loss of water is what kills you. So we need to rehydrate. And luckily for everybody out there, right, oral rehydration therapy is cheap and it's easy. It's just this, right? So a liter of safe water, 6 teaspoons of sugar, and half a teaspoon of salt. That's enough to have people come back from cholera, hydrate them, and then also provide sugar and electrolytes. The salt would be the electrolytes. The sugar provides, yes, it provides some energy for your cells, but it also provides the osmotic draw back into your cells. So we'll take a closer look at the cholera toxin here. So we can see that here's our Vibrio cholera. It's a curved rod. It has a flagella, and it's going to produce a toxin. We've talked about this kind of toxin before, this AB type toxin, where it has like the B is for the binding and the A is the active part. So that's just what it is. When the active part comes into the cell, what it does is it makes the membrane pumps that pump out chloride and bicarbonate. They're going to work really hard to pump out these ions. When these ions go, then we have some positive. You notice how these are negative charged? We have some positive charges following. And this, if you've not taken physiology yet, you'll learn this in physiology. When you do have these particles, these ions come out through a semipermeable membrane like this, it automatically draws water out. So water always follows the solute. This is the pumping out of the solute caused by the cholera toxin. And then we have the water just follows. So in normal diarrhea, your large intestine just kind of moves very quickly. The peristalsis speeds up. And the water loss is basically due to the fact that your large intestines don't have time to reabsorb the water because of that fast peristaltic action. But this is different because this is an osmotic draw out of the intestines. It's much nastier. You can dehydrate much faster this way. So number 6 and number 7 are things to remember about this cholera toxin. So this is a look at what your book has. This is the water that, endemic waters like this can cause Vibrio cholera outbreaks. You also hear about cholera outbreaks when you have like a storm, a hurricane, and it washes water ashore or causes flooding ashore, and that flooding is full of contaminated water. We looked at this before. We have cholera cots where there's a hole through the cot so that it catches the fluids as it leaves the body. And of course, they're going to get intravenous fluids to help rehydrate themselves. And then we have our little guy, our Vibrio cholera. So curved rod, one flagella. So there's another Vibrio out there that is also in the water. It's salt tolerant, and this one's really associated with our shellfish and oysters. So it causes gastroenteritis from raw oysters. And whenever you eat raw food, it's going to be known that you're taking a risk. And so there's nothing really I can say. Like, I eat raw oysters. I think they're delicious, but you just have to live with the fact that you're doing a risky behavior. So this is basically just a food poisoning, right? And it's only going to cause really severe complications if you have an underlying disease or liver disease or diabetes. Okay, so treatment is fluid and electrolyte placement, occasionally antimicrobials, but it's basically just a mild or, some kind of food poisoning. The next one here, Campylobacter jejuni, is another sort of curly rod. It is going to be very common gastroenteritis. So it says the most common bacterial gastroenteritis. It's sensitive to heat. This bacteria can be destroyed in heating. And it does make an enterotoxin that stimulates a secretory diarrhea similar to cholera. So that kind of diarrhea where the electrolytes are lost first. So we can see that Campylobacter jejuni, I really picture this associated with poultry. So cattle, sheep can also be contaminated. And and this is true for all your enteric bacteria, those are in the organs of animals, like to get the bacteria from the inside of the intestines to contaminate the meat, the muscle. It's all contaminated during slaughter. And so raw milk is also a source of this guy. And so if you look at the USDA, just proper heating. So both Salmonella and Campylobacter are pretty wimpy with heat. And so are avian flu viruses. So as long as you cook the poultry to the temperature it says, you'll be fine. The Campylobacter does sometimes make bloody or watery diarrhea, but it doesn't say specifically that it's very active in destroying the membrane. So it's another kind of food poisoning, rehydration, electrolyte balance. And I'll show you this little clip here I have of spreading bacteria in the kitchen, because one of the things that they talked about in the book here with Campylobacter and any meat is that when you wash the meat in the kitchen, you want to be careful about the splash, and you want to be careful about your hands and what your hands.
It is to spread the bug around your kitchen. After a few minutes of quartering the bird under lab conditions, we dimmed the lights to take a look.
Okay, so we turn on the UV lights, we look at the carcass itself, it's heavily contaminated, but then we look at the chopping board and it's sort of contaminated, and you've managed to contaminate the knife very well on the handle. So if you look at your hands, and your hands are covered in the tracer. And then did you touch your face? Oh, there's a small stain there. Yeah, but you can see you've also splashed Campylobacter, or the tracer, onto the chickens that were clean over here.
This explains why we're now told not to wash chicken to avoid splashing bugs around. And as we all know, thorough cooking and washing hands eliminates the risk of food poisoning.
All right, so that was the little video there. Our next bacteria is going to be our Helicobacter pylori, which is actually the same kind of shape as our Campylobacter. It's a curly, curved rod, lots of flagella, and this guy is going to be the cause of most of our stomach ulcers. So you can see that's 90% of our stomach and duodenal ulcers. The duodenum, of course, was the first little section of the small intestine right after the stomach. And a lot of people have this. 2/3, they estimate that the people have the H. pylori in their gut. However, just because you have it in the gut doesn't mean you have infection. You're going to have 20% risk of developing stomach cancer or ulcers. So human stomach and also our mouths, you guys, which means that we can get this through oral kissing and also oral fecal routes. The stomach, as we know, is a very harsh environment. It has a very low pH. So how do we have this normal flora that's able to live in our stomach, right? How can it survive in this acidic environment? The answer's on the next slide. But take a look here at a perfectly healthy colon, and then here we have an ulcer. So remember, an ulcer is an open wound. There's no covering to it. So if you have an ulcer and you have acids produced in the stomach, it's going to really burn. So this is how they do it, right? So Helicobacter pylori burrows underneath the stomach mucus layer. So the stomach will have a very large mucus layer that it uses to protect itself from the acids it makes. So when it digests food, it's not also going to digest the epithelial lining. So what the bacteria do is they burrow under this mucus acid and they find the surface of these epithelial cells, and that's where they're going to stay. And also, in order to combat the acids in the stomach, they do produce an enzyme called urease that does make a basic compound, this nitrogen compound, and it neutralizes ammonia, basically, and it neutralizes our stomach acid. So it does make a little cloud of neutral fluids around itself. And so we can have a little colony of Helicobacter pylori in our stomach, hiding in our mucus and also producing that urease. So because of the presence of the bacteria, right, your immune system is going to be alerted that there's bacteria here, and it's going to cause what you're normally going to find in any infection, inflammation. You're going to have leukocytes coming to the site of infection, and a couple things are going to bother your immune system. The first thing is going to be the bacteria itself, and the second thing is going to be the ammonia that it's producing. So both of those things are going to bother your immune system. The immune system comes in and you have chronic inflammation. Anytime you have chronic inflammation, you're going to have mucosal damage. So you're going to have an area where the mucous membrane has been damaged. It won't be protected by that mucus layer anymore. And you can see that the enzymes and hydrogen ions are able to irritate this area. So that's your ulcer. All right, so that are ulcers. The next bacteria here is Clostridium. Clostridium is going to be anaerobic. So our first true anaerobe, I think. This is going to have 3 species we're going to talk about. Clostridium perfringens is going to be the cause of something called gas gangrene. Clostridium botulinum and Clostridium tetani, they're both going to cause both botulism and tetanus, but they're neurological, so they're going to be in further chapters. However, they're all going to be spore formers, so as you can see the spore here, and they're all going to be anaerobic. So in the question I have here is, what does this mean about infection? It means that when this bacteria does cause infection, it has to be in an anaerobic environment, right? You have to have a very, very low concentration of oxygen. What that means in your body is that the blood supply has to be disrupted, right? So maybe you have a wound where the blood vessels are, not delivering blood to the certain area, and that causes an area to be more oxygen deficient. Or we can have like a puncture wound, some kind of like aseptic puncture. I know aseptic abortions used to be an issue with Clostridium because you have this very low oxygen space and you have a puncture wound that can be, lead to this kind of infection. Also, the anaerobic spaces of our colon, right? Remember, our colon is largely a nice anaerobic environment or very low oxygen, so Clostridium can grow there too. The Clostridium we're going to talk about here in the gut is going to be Clostridioides difficile. I always learned it Clostridium difficile, but I guess now it is Clostridioides difficile. The health community commonly calls it C. diff, and it's a very common infection that is antibiotic-associated colitis. It's also called pseudomembranous colitis. Both of these are considered nosocomial infections because they are usually due to people in the hospital on antibiotics. This is a normal part of our flora in the intestines, and it's sort of naturally antibiotic resistant. So when a person is taking antibiotics, and it's sort of a broad spectrum, it kills a lot of bacteria in your gut, then the Clostridium are naturally resistant to the antibiotic and they can overgrow. And this overgrowth is going to be your antibiotic-associated colitis. So colitis is a term for just inflammation of the colon. Pseudomembranous colitis is because the bacteria will grow in these membranes, and they're not true membranes, they're pseudomembranes. So that's what it is. The greatest risk are for people who are immunocompromised, long hospital stays, older. The antibiotic, of course, are at risk for getting C. diff infection. So lots of different risk factors. The infection is going to be diarrhea. The bad part of this is that you can actually have focal necrosis, which means focal means that there's a specific region. You're not going to have a huge swath of necrosis, but little areas where they ulcerate. And then this is the pseudomembranous colitis, a lot of inflammation in the colon. It can be very bad, cecal perforation. So the cecum is a part of your large intestine, is the first part of the large intestine. And if that perforates, it basically means it breaks open. So that's really bad. So that's basically our Clostridioides difficile infection. And then we're moving on to our Bacillus cereus. Bacillus is a gram-positive rod endospore forming. And the thing with Bacillus cereus, it does make a food poisoning that is very similar to Staph aureus. Okay, so it's an intoxication type food poisoning. However, there's a difference because Bacillus cereus does not come from our body. So remember that Staph aureus was inoculated into the food by the food handler, right? Because Staph aureus is on our skin. So if you were to grate the cheese and then the cheese goes in the food, the Staph aureus from your hand follows the cheese into the food, and that's how the food gets contaminated. In this case, the Bacillus cereus does not come from us, it comes from the environment. So the endospores are already there. Rice is a really big sort of food that Bacillus cereus causes a lot of food poisoning in. So that's why I have it bolded here. And basically, rice will naturally have some of these spores of Bacillus cereus because it's in the soil, right? So when you harvest rice, you're going to have those spores with the rice. When you cook the spores, they are very hardy. Remember, spores can withstand a lot of different temperature extremes. But in this nice warm environment with lots of rice, yum, yum, right? The endospores can germinate. When they germinate, then the Bacillus cereus can go ahead and make that toxin. So illnesses mostly occur after eating the rice or other foods prepared at, and again, temperature abuse for too long. So this is an intoxication very fast, a few hours after ingestion, and you get those food poisoning symptoms. Named after Ceres, the goddess of agriculture. Okay, so these are the bacterial infections of the GI tract. Bacillus cereus, Campylobacter, cholera, C. diff. We didn't cover Perfringens gastroenteritis, but we did cover E. coli. We covered ulcers and Salmonella and Shigella and staph food poisoning and typhoid fever, and we did not cover, I have an X here, for Yersinia, so we didn't cover a couple of those. So let's pause it here, just a nice stop, and then we'll move on to viruses.